Hello everyone. In this stream we will be talking about introduction to medical genetics, a very important subject of an introduction to genetical or medical genetics. Is very important. Let's start by talking about this subject, which could take a long time to finish. It's about 53 slides, very long. I don't know if we can finish it all and understand it fully in this video, but we'll try our best. Let's see here. As we can see here, genetics concerns the process of trait inheritance of parents to offspring. Genetics concerns the process of trait inheritance from parents to offspring, including the molecular structures and function of the genes. This is, of course, important in inheritance, as genetics concerns the process of trait inheritance from parents to offspring, including but not limited to this here. Genetics concerns the process of trait inheritance from parents to offspring, as we can see in this definition. Genetics concerns the process of trait inheritance from parents to offspring. This is offspring, very important inheritance, of course. Next, after this definition, we have more definitions. Talking about the inheritance of genes through offsprings. And here we can point out including as genetics concern the process of transfer from parents to offspring, including the molecular structures and function of genes. This should be highlighted to understand the process of gene inheritance. Here we have the gene behavior, molecular structure and function of the gene is important. Gene behavior in the context of cell or organism. Gene distribution and variation with change in population, as it differ with population and with the distribution of population. It varies during the distribution and variation with change in population. Gene behavior in the context of a cell or organism. We have the molecular structure. Here is the molecular structure and function of genes. As genetics concerns the process of trait inheritance from parents to offspring, including molecular structure and function of genes. Number 1, molecular structure and function of genes. Number 2, gene behavior in the context of traits. In the context of traits or cell organism, gene distribution and variation with change in population. Next, of course, we have the medical genetics. Here we have it. The genome refers to all of the DNA or genetic material in an organism. This is the definition of a genome. Refers to all of the DNA or genetic material in an organism. That's a very important definition here we have, which is called the genome. Next slide. We also have an important structure called the chromosome, which is having the P and the Q and the centromere here in the center. Next slide. we have not this one. This here we have genes are located in the cell nucleus on chromosome. Here we have it. Genes are located in the cell nucleus on chromosomes. This is the location of the gene as it is located in the cell nucleus on the chromosome. Here exactly, cell nucleus on chromosome. These are some chromosomes here. And this is a karyotype. And this is an eukaryotic cell, which is an animal in this case, as we can see in this image. And this one here showing all the types of paired chromosomes. This we should highlight here, the genes are located in the cell chromosome. Very important information. Genes are located in the cell nucleus on a chromosome. Which is here. Next, we can see this human genome containing 23 pairs of chromosomes. The human genome is the total of the genetic information that is held in each human cell. In each human cell, it is usually made up of 46 chromosomes, 22 pairs of autosomes, and one pair of 6 chromosomes. The human genome is the total of genetic information that is held in each human cell. It is usually made up of 46 chromosomes, 22 pairs of autosomes, and one pair of sex chromosome, which are usually X and X for females, and X and Y for males. These are the autosomal chromosomes. We can locate them here. Autosomal chromosome, and this is the male sex chromosome. Next, of course, we will have the animal eukaryotic cell. Animal eukaryotic cell. The animal eukaryotic cell contains about 66 of materials. Or 16 cell organelles. First is the animal plasma cell membrane. This one is the cell membrane, number one. Number two is the smooth endoplasmic reticulum. This is the smooth endoplasmic reticulum. Because it has no ribosomes on its edges. Three, we have the vacuoles. This is number three, the vacuoles, used to phagocyte liquids. Number four is the Golgi body and vessels, which is here. This one is a Golgi body, and this one also could be called a Golgi body. This one is also a Golgi body, also containing ribosomes and digestive materials. Number five, the mitochondria. As we can see here, many mitochondria, 1, 2, 3 mitochondria, 4, 5, and 6 mitochondria. Then we have the ribosomes, number 6, which are on all over the eukaryotic cell, especially on the rough endoplasmic reticulum. Rough endoplasmic reticulum. Number 7, we have the centrosols or centrioles. What is number 7? I cannot find it in this slide. Maybe it is hiding somewhere. This is number 6, the ribosomes, but number 7 is nowhere to be found in this picture of animal eukaryotic cell, but it is mostly a centriole that is between this 1 and 2 chromosomes. So it should be here, number 7. Maybe this one. Next, number 8 is rough endoplasmic reticulum. The rough endoplasmic reticulum, as we can see, surrounded by ribosomes. Lots and lots of ribosomes are here, and ribosomes are all over the place. Here, vacuoles, mitochondria, Golgi apparatus, and this is number 7, centrioles. Oh, this is the centrioles. We were looking for the centrioles and we found them. Of course, they are important in the process of multiplying of the cell. It can be, or it can go for both sides of the chromosome and drag them together. Very important part of the cell. And cell reproduction process. Number 9 is the double nuclear envelope. Double nuclear envelope surrounding the nucleus material, nucleoplasm, the double nuclear envelope, which is, as we can see here, double and surrounding the nucleoplasm, surrounding the chromosomes, and it is very important in the cell protection, nucleoplasm protection, and the protection of and exchanging of nutrition between outside the nucleus or the nucleolus and the chromosomes and inside the nucleolus or the nucleoplasm with the cytoplasm of the cell. Next, we have the nucleolus within the double nuclear envelope. We can see the nucleolus, just like the cytoplasm, mitochondria, but this one is the nucleolus, important within cell, providing protein and within the process of multiplying and making more cell. Number 12, this was number 10, nucleolus, and number 12 here is the nuclear pore. The nuclear pores are small pins or small holes within the double envelope, double nuclear envelope, are small pores called the nuclear pores. Nuclear pores. Here we can draw them, maybe, just to show up how many of nuclear pores are within the envelope, the nuclear envelope. This is 1, 2, 3, 4, 5, 6 pore, 7, 8, and 9. Maybe even 10 within this picture. These are the nuclear pores. Number 12. Number 13 is the nucleoplasm. Nucleoplasm. Here, all over this place, we have the nucleoplasm surrounding the nucleolus and the chromosomes within the pores of the double nuclear envelope. We have the nucleoplasm, next let's just erase this. We don't need the pores. Next we have one second. The cilium here, the cilium is number 14, showing a projection coming in from the cell membrane. This one here is the cilium, maybe just like the flagella and the cilia, which are important in sometimes movement, sometimes just sticking to objects. The cilium. Next organelle is the basal body, which is number 15. The basal body here used to provide the cilium with the force needed to pivot and hold the cilium in its place, called the basal body. We can show it here in this dark area. It's called the basal body or the basal membrane. Next we have the cytoskeleton. The cytoskeleton here, number 16, shown in the picture or the image here in the animal eukaryotic cell. We can see the cytoskeleton. Not very defined in this picture, but it is mostly used to maintain the shape of the plasma, the circular shape, and maintain the strength of the shape of the animal eukaryotic cell. I think we should move to the next slide because we've already talked enough about this one. Well, in this slide we have the basic concepts of heredity. The basic concepts of heredity. We have two paragraphs. Genes provide instruction for the whole human, traits including physical features and how body parts function. This is the importance of the gene. Each person inherits a particular mix of maternal and paternal genes. Very easy to understand about genes providing heredity and the basic concepts of heredity of the providing for all traits of human, physical features, how body parts function, and each person inherits paternal and maternal genes. The next slide, we can see here the basic concepts of heredity continues. In saying the gene or the genes of the human body, genes have about 25 or 21,500. You can see it here. Human have 21,500 genes. Also, the chemical instruction for building proteins are the genes. The building or the chemical instructions for building proteins they possess. Locus, a specific location on a chromosome. This is the location on a chromosome called the locus, but mostly genes possess the chemical instruction for building a protein. And the human having 21,500, very important number, 21,500 genes. Next is the diploid cells. The diploid cells contain 2 copies of each genes on pairs of homologous chromosomes. These are the diploid cells containing 2 chromosomes or 2 pairs of each gene on a pair of chromosomes. We can draw this diploid cell just to remember it. It would be something like this, and it contains 2 copies of each gene. This is one gene, and this is another gene. This one and this one. Contains 2 copies of each cells on each gene, pairs of homologous chromosomes. This chromosome is homologous to this chromosome, containing a diploid, which are called the diploid cells. Next, we have the allele. The allele is each version of a gene. This is a version, this is another version of a gene called the allele. Let's move to the next slide. We have here many genetic traits having dominant and recessive forms. We have many people here having many genetic traits. genetic trait having dominant and recessive forms. And of course, the dominant forms just become the new person. Basic concepts of heredity. These are the homogeneous condition, the heterogeneous condition, and the dominant. These are the basic concepts of heredity. The basic concepts of heredity. We can show up here, show the hemo or homozygous condition, the heterozygous condition, and the dominant allele. The homozygous condition, which is identical alleles, the identical alleles is a homogeneous or a homozygous condition. There is a heterozygous condition with different alleles. This here we have different alleles, and before that we have identical alleles. Homozygotes or homozygous, then the heterozygous. The heterozygous. The dominant allele being the effect that masks recessive allele paired with. Let's say that we have like two alleles in one chromosome. One allele is here, the second is here. This one is dominant, and this one is recessive. Then we can say that this is a heterogeneous condition. If we have one is here is dominant, the other is dominant here, then this is a homogeneous or a homozygous condition. Homozygous condition. And of course the dominant allele will mask the recessive allele paired with it. Masks here, we can write masks the recessive allele paired with it, and we'll delete it. So the dominant will stay and the recessive will be masked. Let's move to the next slide now. We have the basic concepts of heredity. Basic concepts of heredity. As we can see, we have genetic representation of the recessive and dominant alleles. This is a homozygous, and this is a heterozygous, as we discussed before. Oh, this one is also a homozygous with the recessive allele. This one is a heterozygous with a dominant one and recessive alleles. So this one is heterozygous with dominant and recessive. with a dominant and recessive. Alleles. We can point out which one is the recessive and which one is the not recessive. 1 second. So here we have the recessive allele, and here we have the dominant allele. This one is dominant, and this one is recessive. The dominant and the recessive alleles. And that's why it is a heterozygous, not a homozygous. And of course this one is a dominant homozygous. Recessive homozygous. Heterogeneous, not recessive and not dominant heterozygous. It is only heterozygous, and of course it is going to be taking the physical form of the dominant allele. Next, we will be having the genotype here, inherited alleles. Genotype is the inherited alleles, and the phenotype is the observational, functional, or physical traits. So there is a difference between genotype and phenotype, as the genotype contains many recessive, this is small a could be here, and the phenotype is only showing the dominant physical traits, functional and observable. That's why it's a phenotype, not a genotype. Moving to the next slide, we have the medical genetics here, as we can see, and medical genetics is a specialty of medicine which we will discuss, and we have discussed before, and it was the main subject of this lecture. It's a specialty of medicine that involves the diagnosis and management of heredity and disorders. So what is the definition of medical genetics, or the specialty of medicine that is involved with diagnosis and management of heredity, which is the transfer of alleles from the parents to the newborn, or to the child, to the next generation, and disorders which could happen while the transfer process between parents and the new generation. Again, one more time, the medical specialty of medicine that is involved with diagnosis and management of heredity and disorders. Heredity and disorders. Next slide is this one. This is the next slide, talking about what is medical genetics. What is medical genetics? As a result of a rapid progress in molecular genetics, gene therapy, the insertion of the normal genes into patients in order to correct the genetic such normal into patient in order to correct genetic disease is now possible. This is talking about the result of rapid progress, just because in medical science that is developing so fast, we can now say that genetic engineering is possible, and we can read this one more time. As a result of rapid progress in molecular genetics, genetic therapy, and gene therapy, the insertion of the normal genes into patients in order to correct genetic disease is now possible. Next paragraph we will be talking about, let's highlight this one, just to remember it, because it's a very advanced information. As a result of rapid progress in the molecular genetics, gene therapy, we can say that it is possible for genetic engineering to be happen in this time. Here we can see medical genetics, and next slide, the medical genetics we have studied before, involving application of genes, inheritance, mapping disease genes specific location on chromosome, analyzing the molecular mechanism through which cause disease, diagnosis and treatment of genetic disease and disorders. We haven't studied this very in depth and immersively, so we can just highlight it in order to study it again. We can say here studies, what is medical genetics? Medical genetics involve any application of genetic in medical practice. It thus include studies of inheritance of disease in families, mapping a disease gene to specific location on chromosomes, analysis of molecular mechanisms through which genes cause diseases, and diagnosis and treatment of genetic diseases in general. In family and analyzing the molecular mechanism through which the gene causes a disease. Molecular mechanic, mapping the disease gene, etc. Studying the inheritance in families and diagnosis and treatment of genetic disease. On this slide we were just talking about, as a result of a rapid progress in molecular genetic gene therapy, the insertion of the normal genes into patients in order to correct genetic diseases is not possible. Next paragraph, we'll be talking about medical genetics, that is also includes medical genetics, that also include genetic consulting, counseling, which involve the communication of information regarding risks, progresses, and treatment for patients and their families. This we can say very important also. We can highlight it to just remember the medical genetics information also include genetic counseling, which involves the communication of information regarding risks, prognosis, and treatments for patients and their families. So counseling is also a field in medical genetic, just like medical therapy. Next slide, we have the diseases, the genetic diseases, very important subject. So what is a genetic disorder? What is a genetic disorder? Simply, a genetic disorder is a disease that is caused by an abnormality in individual's DNA. This is a genetic disease. What is a genetic disorder? Simply, genetic disorder is a disease that is caused by an abnormal individual's DNA, or an abnormality in an individual's DNA. And abnormality can range from a small mutation. This is a small mutation, this word is important, mutation, in a single gene, to addition or subtraction of an entire chromosome. So it is ranging from a single gene, small mutation, or subtraction of an entire chromosome or set of chromosomes. And this subtraction and addition is also important in medical genetics. Medical genetics. This is the abnormalities in genetic disorder. What is genetic disorder? A genetic disorder is a disease that is caused by abnormality in an individual DNA. What is these abnormalities? These can range from a small mutation in a single gene to addition or subtraction for an entire chromosome or set of chromosomes. Next slide. We will be talking about or discussing this genetic disease. This slide is dedicated to genetic diseases. Characteristics of genetic disorders. What are the characteristics of the genetic disorders? These contain the congenital, the mode of inheritance, the population distribution, the familia, or part of characteristic of gene disorders. Congenital mode of inheritance, population distribution, familia. I don't get what is familia, but it's probably a family thing. Congenital is from the newborn, and of course the mode of inheritance, population, and distribution are easier to understand. Next slide is genetic disease, classification of genetic disorders. Single gene disorder, chromosome disorder, multifactorial disorder, somatic cell genetic disorder. This contains the body, mitochondrial disorders. These are all classes or characteristics and classes and classification of genetic disorders. Single gene, chromosome, multifactorial, somatic cell genetic, mitochondrial, all disorders classification. Next slide. We have the genetic disease. Here the genetic disease, single gene disorder, which is one classification that we have already talked about, the single gene disorder. This single gene disorder results when a mutation causes the protein product of a single gene to be altered or missing. It results from a mutation, causes the protein product of a single gene to be altered or missing. This was a single gene mutation, which contained the cystic fibrosis, the Fragile syndrome, the sickle cell disease. All are causes from single gene mutation. And we know that the sickle cell is going to be affecting the blood cells, causing anemia. Then the Fragile syndrome is just a fragile syndrome, this weak, fragile syndrome. The people will become weak or weaker. Then we have the cystic fibrosis. Cyst will be formulated with many, which could cause fibrosis in the human body. Next, we have medical genetics. The medical genetics and cystic fibrosis. Medical genetics and cystic fibrosis, there are three nucleotide nucleotide deletion from the coding sequence. This is a single gene classification problem within the genes. When there is a 3 nucleotide deletion, 3 nucleotide deletion from the coding sequence, which will cause a deletion of phenylalanine, This phenylalanine is causing the cystic fibrosis. The phenylalanine deletion will cause cystic fibrosis. And there are 3 nucleotide deletion from the coding sequence, causing deletion of phenylalanine. This picture showing the cystic fibrosis, which is a hereditary disorder characterized by lung congestion and infection and malabsorption of nutrients by the pancreas. Here we can see in the lungs, cystic fibrosis, hereditary disorder characterized by lung congestion, infection and malabsorption of nutrients by pancreas. Very important classification is the cystic fibrosis. The cystic fibrosis. And the phenylalanine is related to the cystic fibrosis. Next we have, in chromosome disorders, entire chromosomes or large segments of them are missing, duplicated, or otherwise altered. This is chromosome disorders. It is one classification that we already discussed, which was not a single gene disorder, it was chromosome disorder. Then we will be talking about multifactorial, somatic, genetic, mitochondrial. So this one is a chromosome problem, which is a disorder that an entire chromosome or large segments of them are missing, duplicated, or otherwise altered. Here we have an example for the chromosome syndrome problem. Chromosome deletion, the trisomy 13, the presence of an extra third chromosome 13 in all of these cells. As we can see, this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. There is an extra chromosome here in the Pato syndrome. Pato syndrome, very serious syndrome, causing a third chromosome in the 13th cell of chromosomes, of all cells in the body, called the Plato syndrome. We have discussed before the phenylalanine. Cystic fibrosis is another serious syndrome which was caused by a mutation, not in one gene. It's actually not in one chromosome, but in a single gene, the cystic fibrosis. And in a single chromosome addition is the Plato syndrome. And we have the diagnosis and treatment of the Plato syndrome. Diagnosis, but there is no treatment for the Plato syndrome, or the Pato syndrome. Pato syndrome. Even in cystic fibrosis, there is no possible or easy solution and treatment for the cystic fibrosis. which is caused by the phenylalanine, due to a disorder in the gene, and this one is a disorder in the chromosome. There is no treatment, but can be easily diagnosed. Genetic disorder, multifactorial disorder, this is the next classification of disorders in genetics, result from mutation in multiple genes, often coupled with environmental causes. So multifactorial disorders results from mutation in multiple genes, often coupled with environmental causes. These environmental causes, the food, and it's a multifactorial disorder. Multifactorial mutation in multiple genes. Multifactorial disorder, multiple genes, coupled with environmental problems or causes. Here we can say the identified obesity gene. Here we can identify it, causing the obesity with genetic problems in chromosome 2. Unidentified obesity gene, here too, we can see it on chromosome 5 and chromosome 8 and chromosome 18. How do genes interact with environmental factors? Of course, with the food being accessible, the identified obesity gene, the multifactorial gene disorder, resulting within multiple genes, as we saw in 8 and 18, with environmental problems. 2, 5, 8, and 18 identified obesity gene. Next, we have how fingerprints work. This is another subject with fingerprints. The polygenic trait, simple loop, double loop, wall and arch. Polygenic trait. Dermatoglyptics, ridges on finger, palm, toes, and feet are all fingerprints related to genetics, influenced by prenatal environment, nutrition of the mother, rate of finger formation and growth, are all environmental factors. Multifactorial disorder or multifactorial causes the fingerprint to look how it looks from the nourishment from the mother and finger formation, which are environmental multifactorial gene related dermatoglyptics, which could appear on the feet, finger, palm, toes, and many other ridges, is a polygenic trait. It is not multifactorial, it is a polygenic trait, not like the sickle cells or fragile X syndrome, cystic fibrosis caused by single gene disorder, but it is a multifactorial disorder. Let's move to the next slide. We have other multifactorial disorders like diabetes, hypertension, high blood pressure, heart-related problems, arthritis, Alzheimer's disease, or even different cancers and breast cancers. As we can see here, people suffering from cancers and diabetes and high blood pressure, asthmatic, Alzheimer here, many other heart-related problems like blood pressure. Cancer could occur also from genes. Next, we have genetic disease, which is the main subject of medical genetics. Somatic, this is not a multifactorial, it's somatic cell genetic cell disease. Somatic cell genetic disease. Result from altered genetic materials in somatic cells. Results from the altered genetic material in somatic cells. Very important results. Altered genetic material in somatic cells like cancer. Cancer is a multifactorial, you can say a somatic cell genetic disorder, not multifactorial in this case. Next slide. We have the genetic disease, mitochondrial genetic disease. The mitochondrial we have discussed in the previous episode, previous slides. After the somatic is the mitochondrial disorder. The mitochondrial disorder after a single gene disorder, chromosome disorder, multifactorial disorder, somatic and mitochondrial. This time we will talk about the mitochondrial disorders. I don't think we'll be finishing all this lecture. We will finish half of it in this live stream, and other live stream we'll be finishing the other half of the medical genetics lecture. Mitochondrial genetic diseases due to the mutation of mitochondrial DNA, like Leber heredity, optic neuropathy, loss of central vision. All are mutation in the mitochondrial DNA. Examples: Leber heredity, optic Leber heredity optic disorder or disease. This one here. Let's highlight it. Leber heredity optic neuropathy loss of central optic vision. This was a mitochondrial genetic disease. Then we have chromosomal abnormalities. This is another subject talking about common cytogenic diseases. Common cytogenic diseases. Chromosomal abnormality is like it is a structural numerical abnormality of chromosomes involving autosomes or sex chromosomes. Structural numerical abnormality of chromosomes involving autosomal or sex chromosomes, common cytogenic diseases. Chromosomal abnormalities. Also abnormalities in autosomal and sex chromosomes. There are types, classification of these autosomal and sex chromosomes abnormalities. We have numerical abnormalities and structural abnormalities of chromosomal abnormalities. Let's see what it is about. Numerical abnormality. And structural abnormality. Beginning with the numerical abnormality. It involves the gain or loss of one or more chromosomes, known as aneuploid, or addition of one or more complete haploid complements known as polyploid or euploid. So a loss of one chromosome is aneuploid. Addition of one or more haploid complements is polyploid or euploid. So we have aneuploid, polyploid, then we have the aneuploid. Starting with the aneuploid, which is the loss of one or more chromosome, we have the trisomy, the monosomy, and the tetrasomy. The trisomy having 3 copies of a chromosome instead of normal 2, which is very abnormal. Example, trisomy 21, Down syndrome, 47, XX plus 21 or 47XY plus 21. This trisomy 21 in Down syndrome, losing one chromosome instead of having the usual pairs of the 23 pairs, we have now only 21, which is by multiplying becoming 42, not 46. In Down syndrome we have 47, XX plus 21, or 47, which is abnormal. Then we have XY plus 21. Having 3 copies of chromosomes instead of normal 2. In monosomy, 2n minus 1, absence of one chromosome is also the monosome is a part of aneuploid, and the trisomy is also part of aneuploid. The monosomy, absence of one chromosome from pair, example, monosomy X, Turner syndrome, 45X or 45X0. That's a monosomy, the absence of one chromosome from a pair. Example, monosomy Turner syndrome. This one we have 45 instead of the normal 46 or 45 here with X0 or with X. We have the tetrasomy or the tetrasomy, polysomy, 2n plus 2, having 4 copies of chromosomes instead of a normal 2. So here we have the 48, X, 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 and another X. These are 4 copies of chromosomes instead of a normal XX chromosome. Plus it is a 48 here, which is a tetrasomy. And here we before talked about the XX plus 21. These are all variation in chromosome in human, trisomy, monosomy, tetrasomy, polysomy. Polysomy we have no example, but in monosomy we have the example of Turner syndrome, and in trisomy we have the example of Down syndrome. All part of aneuploids, which is different from the human 23 pairs of chromosome or 46 chromosomes. These numerical abnormalities, I think we will be discussing then the polyploid ploid, and then we will talk about the euploid. This is also about aneuploid. Results from non-adjusting at meiosis. There's an explanation why there is an aneuploid. An explanation saying that there is no adjunction at meiosis or mitosis. What is non-adjunction? Now we will explain what is there is, why there is non-adjunction and what is the non-adjunction during meiosis, mitosis. Failure of paired chromosomes or sister chromosomes to pass to opposite poles of the spindle at the anaphase or of mitosis or mitosis. So it's a failure of paired chromosomes or sister chromatid. Sister chromatid, like here we have a sister chromatid from passing to opposite poles. After they get separated, they cannot be passed to opposite poles of the spindle at the synaptic or at mitosis or meiosis. The causes of non-junction or non-disjunction. This is non-disjunction. What is the non-disjunction? We have already explained that it's a failure of chromosome or sister chromatid to pass opposite side of mitosis and meiosis. Now we will talk about causes of non-adjunction, and this was just a definition. The causes of non-adjunction is advanced maternal age, radiation, delayed fertilization, chemicals and genetic predisposing factors are all causes of this non-adjunction. Let's move to the next slide. There's genetic predisposition. Also, predisposition is another cause of a non-adjunction. Now this slide will be talking about numerical abnormalities, continuing with the polyploid addition of one or more complete haploid components. Addition of one or more complete haploid components, multiplies of haploid number which are higher than diploid, example triploid, tetraploid, and so on. We've already talked about the missing of one chromosome. This time we'll talk about addition of one chromosome. Addition. Polyploid, addition of one or more complete or multiplies haploid number that are higher than diploid, example triploid 3n, tetraploid 4n, and so on. Of this ploid numerical abnormality. Next slide. We'll be talking about how many types of trisomy aneuploids Of course, there is aneuploid of autosomes trisomy, which is addition to a chromosome. Many types of autosomal aneuploids have been found in spontaneous abortion, but only trisomic condition of some of these smaller chromosomes are compatible with past natural life, unless abnormal cell line occur in meiotic form, which a normal cell line. 3 autosomal trisomes are well established in man. So the Down syndrome is an autosome or trisomy, the aneuploid trisomy. Edwards syndrome and trisomy Patau's syndrome. I think we already talked about Down and the trisomy 21, trisomy 13, trisomy 18. All three types show an increased mean in maternal age at birth. So they show an increased maternal age at birth. Okay. Let's read this again, maybe you'll understand this time. Many types of autosomal aneuploids have been found in spontaneous abortion, but only trismus or trisomic condition of some of the smaller autosomes are compatible with postnatal life unless the abnormal cell line occur in mosaic form with normal cell lines, 3 autosomes. I cannot go back and see what is the autosome because it's been very far away, trisomy. We should review this later. The trisomy subject we've already talked about, but I don't think we can just go back and see what we read and relate it to what we are reading right now. Anyways, the aneuploid of autosomes trisomy. More talking about trisomy 21, 47XX 21 or 47XY 21, Down syndrome. Down syndrome is the most common autosomal abnormality in men, occurring with a frequency of about 1 in 800 living birth. Males are more affected than females. We were talking about this here. Now we will talk the increased incidence of Down syndrome with increasing maternal age is quite dramatic. As we can see, 1 in 1,000 after 30 and 1 in 100 after 30, 40 years old age women will cause Down syndrome. 1 in 50 at age of 45, which is very dangerous age for the Down syndrome. That's why women should marry at a younger age to avoid the Down syndrome, which could occur in this age. Very dangerous to or for a woman to marry at an older age. As we saw here, at 45 it could cause Down syndrome 150. At 40 she could cause Down syndrome 1 to 100. At 30 it is better in 1 in 1,000. So 30 would be a good time to marry. And age 20 would be very good time to marry in relation to Down syndrome. Next, See this here. Just cleaning the page. And let's move on to the next slide. We have these are chromosomes, an additional chromosome and chromosome number 21. As we discussed in the page here, there is an additional chromosome, and 21 there is a 47XX plus 47XX plus 21, or 47XY additional to the 21 pair of chromosomes. Here we can see them in the other slide very clearly. In the 21 pair we can see an additional chromosome, making it not 46 but a 47 number of chromosome with an additional chromosome in each cell in human body. Here we can see a flattened nose of Down syndrome. Let's point this flattened nose and face upward, staining eyes, as we can see here, upward slanting eyes, upward slanting eyes here, then flattened head or flattened face, as we can see, upward eyes, upward slanting eyes here, and the flattened nose, as we can see. Then in this next description about legs, we have widely separated first and second toes. First and second toes widely separated. There is a wide distance here between these two fingers, about 2 centimeters, let's say, or even more, and increased skin creases, At last we can say option. Let's finish this writing here. Single palmar, as we can see, single palmar crease here, short fifth finger that curves inward. I don't know which one this one is talking about, but the fifth finger is curving inward, which is a very special mark or trait with Down's syndrome. Let's move to the next slide now. Not all cases of Down syndrome related to maternal age effects. Not all cases of Down syndrome related to maternal age effects. This is very important. Some may be results from non-adjunction in the father, or result from non-adjunction occur because of other causes of non-adjunction. Lifespan, paternal diagnosis, flathead, etc. The lifespan of Down syndrome, it means the age is 16 years. The mean age is 16 years, though it varies from few weeks to decades. Paternal diagnosis of Down syndrome can be made by chromosomal analysis, chorionic villi and amniotic fluid cells. Then we have this. The clinical features involved are flat board face, which already we have discussed. Then we have epicanthus, a skin fold in the middle angle of the eye. Then a nose which has a low bridge. Next slide, we have the aneuploid of autosomes. Clinical features here. The tongue usually protrudes is forward, lacking a central fissure. Small ears. Hands are short and broad. They may be, this is all because of the Down's syndrome. There may be seminar or simian crease angle, palmar causing or palmar crease resulting because of a fusion of heart and head lines. There is often a wedge gap between the first and second toe of the feet. There is often a wedge or a gap between the first and second toe of the feet, which we already saw before, that there is a gap in the feet, fingers. Then we have next, approximately 1/3 of patients suffer from major heart defects, hyperflexibility of the joints, then irregular and abnormal sets of teeth. Other clinical features of the Down syndrome includes mental retardation, IQ scores between 25 to 50, patient credited with being good nature and asphyxiation, with having a sense of rhythm, or sense to rhythm, which is a good thing, of course. The aneuploid of outcomes, trisomy 18, which is called also Edwards syndrome. In the patient will have the 47 chromosome, 23 pair plus 1 chromosome, male, plus 18. And the 18 we can see here having another chromosome. And female, also in the chromosome pair number 18, the patient will be having an additional chromosome. causing the number of chromosomes to rise from the normal 46 to 47 chromosomes. Other chromosome will not be affected. Sexual chromosome, autosomal chromosome will be the same, or the same number. These are the autosomes, and these are the sex chromosomes, will be the same number. So to the next slide. The next disease or next genetic problem, mutation, aneuploid of sex chromosome. Klinefelter syndrome or Klinefelter syndrome. And this sex chromosome having another Y chromosome in addition to the two X chromosomes. In this case, a female having an additional Y chromosome. And the sex pairs of chromosome. And of course, the autosomal chromosome will not be affected. 22 autosomal and 1, the 23rd. Sex chromosome will suffer from the Klinefelter syndrome. Next is the Turner syndrome of how many chromosomes? 45. Aneuploid of sex chromosome 45. X0, that means we are lacking a chromosome, which should be XX or XY, but in this case it is only X0. The Turner syndrome occurs in 1 per 5,000 newborn females. Missing one X chromosome most cases, and mosaicism, the mosaicism isochromosome, ring chromosome and deleting of X chromosome was observed. The majority of Turner syndrome are lost as spontaneous abortion. The clinical features is a short structure and widowed neck and variety of skeletal abnormalities. The Turner syndrome also, as we can see in this picture, there is a problem in the chromosome missing from the sex chromosome here, which is X0, female. This is here called sex chromosome. And these are the autosomal chromosome. Autosomal chromosome, which are intact and doesn't appear to have any difference. We can see is problems of Turner's syndrome. Short structure, low hairline, fold skin of neck, constriction of aorta, undeveloped breasts, shield-shaped thorax, elbow deformity, etc. Rudimentary ovaries. Then we have no menstruation. Short and metacaliber IV, small fingernails, brown spots of navel on skin. The main being the short structure is a very distinct feature of the Turner syndrome. Next, we have the aneuploid of sex chromosome. This is also Turner syndrome. Clinical features: replacement of ovaries by streak, gonads, and sequence. There is amenorrhea, immature sex development, and serology. Intelligence is generally within the normal range, but maybe 15 to 10 points lower than siblings. Edema of the hands and feet, and others. Mostly, of course, being the short structure. This is the most important feature. Ovaries, and the good thing is that it's only 15 to 10 reduce in IQ and lower of thinking and smartness than the other siblings. Here we have the aneuploid. Of 6 chromosomes. Other syndromes with multiple 6 chromosomes, usual 4 more 6 chromosomes in the karyotype, have been described, but are very rare. This one, 48, with the 6 chromosome having additional 2 in order to be 46 plus 2, this is 48 chromosome here. This one is additional 3 to the 46, causing a 49, or there are many variations, here being an additional 3, also 49, but with Y or YY and XY, etc. General number of sex chromosomes associated with a greater maternal retardation and physical malformities. The greater the number is, as we can see here, 6 chromosome with additional 3, here's additional 2, and here's additional 2. But this is when it's additional 3, there is a greater number of mental retardation and physical malformation. Found in pure and Mosaican. Found in pure and Mosaican. Next slide. Mosaican. We have the chromosomal abnormalities, structural abnormalities. Let's start with the structural abnormalities. It results from chromosome breakage, followed by reunion of the broken ends in different configuration. The factors responsible for structural abnormalities are mainly ionizing radiation, chemical agents, and viral infections. These are the factors responsible for structural abnormalities: the ionizing radiation, chemical agents, and viral infections. Chemical, viral, and physical. Moving to the next slide, with the continuing structural abnormalities. involved two main types. The balanced arrangement, which also includes two types. The exchange or arrangement of genetic material does not cause any missing or external genetic material. Balanced arrangements are generally harmless, with the exception of some cases in which one of the breakpoints damage an important functional gene. An important functional gene, if it's breaking at this point, it can cause damage, as we can write it here. Then balanced arrangement carriers are often at risk of producing children with unbalanced chromosomal complements. Balanced arrangements are often at risk of producing children with unbalanced chromosomal complements. Then we go to the structural abnormality unbalanced arrangements, in which the chromosomal complement contains an incorrect amount of chromosomal material, usually associated with very severe clinical effects. So this one is severe clinical effects is the unbalanced arrangement structural abnormality. This slide showing us how serious it is the structural abnormality could be. Type of structural abnormality, deletion, loss of genetic material results in monosomy for this segment of chromosome said usually large deletion will be incompatible with the survive to term. Terminal deletion and insertional deletion. Terminal deletion involves a single break of terminal part or chromosome, and the broken part is subsequently lost. This is the terminal one. The interstitial deletion involves two breaks, and the intervening portion of the chromosome is lost. This one is terminal, and this one is interstitial. With the interstitial, of course, being more harmful, as we can see here, because it breaks in two parts, and the terminal only break in one part here. And this portion of chromosome here is lost, which is a larger portion than this one and the terminal one. Let's move to the next slide now. With the structural abnormalities, we will be having terminal deletion, the Cre-Du-Chat syndrome. This is a very serious syndrome in structural abnormality. Here. With 46 chromosomes in the female, 5p or 46, which is the normal number of chromosomes, with the male, and the 5p indicating a Cre-Du-Chat syndrome. It results from deletion of short arm of chromosome number 5. This is an important number here, with the Cri-du-Chat syndrome. The incidence is 1 per 50,000 births. The clinical features are mewing cry of affecting babies, which disappear at first month of infancy. Microcephaly, white set eyes, epicanthus, low set malformed ears, severe mental retardation. It's called the Cri-du-Chat syndrome, only 1 in 50,000 births. Structural abnormalities. In structural abnormalities, we have deletion. Some deletion occur as microdeletion, which cannot be detected by microscope. It is needed for high-resolution cytogenetics. Molecular cytogenesis, fish or molecular genesis analyzes PCR. And we have the parade, the Prader, Will, and Angelman syndrome. The Angelman syndrome, Prader, Will, analysis PCR, fish, all part of the deletion structural abnormality. Next, within structural abnormality, we have the duplication and inversion. Some genetic material double, and some involve to break along the chromosome, which given segment is reversed in position. Pericentric invasion, paracentric invasion. The pericentric invasion involves both QP arms, which centron forms, and paracentric invert segments involve only the arm. This is paracentric and this is pericentric. As we can see in the pericentric, the D change in place here with the C and B. As it was first the D, then C, then B, it becomes first B, then the C stays the same place, and D change between it and between D. But in paracentric inversion, we can see only two chromosomes exchange places. The D and C1, this one is the D, and this is the C. We can see here it becomes first the C, then the D, paracentric involving 2, pericentric involving 3 chromosomes. Same one involving only one arm, paracentric involving one arm, and the pericentric involving both PQ arm with a centromere. Okay, this is this is a duplicated one. Maternal doubled. Some genetic material could get doubled here, as we can see. Let's move to the next slide then. These are all structural abnormalities. More structural abnormalities are within the ring chromosome, which is a loss of telomeres or ends of both arms of chromosome and formation of a ring structure. Formation of a ring structure. Very important disease or very important abnormality we are having. The ring chromosomes of telomeres, here those of telomeres, arm of chromosome formation of a ring structure. This is the ring structure, this is the ring chromosome structural abnormality. Isochromosome, creation of two non-identical chromosomes here. One is a combination of the two short arms here. The other is a combination of two long arms, new chromosome in which two arms genetically similar. This is the transverse break at centromere and formation of ABB. This one is ABCD, this one is ED, CC and D, with the union of centromic elements of the sister chromosome, causing one that will lose and the other remaining. Next, of structural abnormalities, the insertion. Insertion, no loss of genetic material, but arrangement of genetic material to non-homologous chromosomes result from two breaks of one chromosome release a fragment, and one break in another chromosome to insert this fragment. As we can see here, there is an insertion of this fragment to this chromosome, causing this to lose its genetical material, and this to gain new material. Next, of structural abnormalities. Let's see how many pages or how many slides we have. About 2 to 3. The translocation. It involves exchange of genetic material between two chromosomes involving two types. This is translocation. Involves exchange of genetic material between two chromosomes, structural abnormality involving reciprocal translocation. It says here no loss of genetic material, but an exchange of genetic material between two non-homologous chromosomes to form two derivative chromosomes. This one with this one going to be an exchange in genetic material. The white chromosome gives here to the black one, and there is an exchange. This will be called translocation. On translocation, this is reciprocal. There is another one called Robertsian Robertsonian translocation, centric fusion. This one here, this two pair of chromosomes. Then we have this one is broken. The other one is also broken. The new chromosome will contain this part, and the small broken part from here, and the long chromosome will manage to one second. Will manage to be with the other one, or to form with the other one here, and this one will form together and will make this very long chromosome. And of course these two have made this one, which is called Robertsonian translocation centric fusion. It results from break at one at or near the centromere into acrocentric chromosomes DD, DG, and GG, and subsequent fusion of their long arms. The fragment formed by fusion of their short arms is lost. It is a functionally balanced translocation through chromosome number is reduced to 45. Carriers are normal because of two short arms that lost do not carry important genetic material. These are lost and they do not carry genetic material, but those big do carry and they survive. The Robertson or the Robertsonian translocation is better than the normal translocation or the reciprocal translocation, which includes no loss of genetic material, but an exchange in material between two homologous chromosomes to form the derivative hormone. Next, so I think we studied all the inversion, the structural abnormalities. We finished this lecture. Just a quick review to the lecture. We can say that we have studied the structural abnormalities, insertion. This one was insertion, structural abnormality. We studied many others like ring chromosome, isochromosome, also part of structural abnormality with many different shapes, duplication, inversion. Here we studied the inversion, polycentric, paracentric. This was the end of this genetic material lecture. We have the structural abnormalities. We have to study this very carefully next time. PCR, very important diseases that we'll be studying in depth in the future, hopefully. Then we have structural abnormalities, terminal deletion. Also this chat, Cry-Due syndrome, very low survival rate, very low survival rate, 1 in 50,000. We study the types of structural abnormalities, the deletion, which was the first structural abnormality that we studied, of this one getting loss and this one getting loss too, with the intra-stitial deletion. Very long lecture actually. This is just a review. Structural abnormalities, unbalanced arrangement. We have the unbalanced arrangement, severe chromosome effects, clinical material is usually associated with very severe clinical effects. The unbalanced structural abnormality, I think this one is unbalanced, but I don't know if it causes severe or dangerous clinical effects. Next, we did study or previously we studied the balance arrangements. Structural analysis involved two main types: genetic exchange of arrangements. Important functional genes, which could be damaged, of the balance arrangement, are generally harmless. We did study the ionization, radiation, chemical agents, and variation, and viral infection, which could cause structural abnormalities. And we studied the structural abnormalities one by one. One second. We did study the X and Y aneuploid. Only aneuploid we studied of sex chromosomes. The syndrome would multiply sex chromosomes with other symptoms of aneuploid. This is an aneuploid of sex chromosome with X0, 45 chromosome, missing sexual chromosome. This was the Turner syndrome, of course, which was a very serious illness, but does not affect the intelligence of the kid. Just make it shorter, or make the human shorter. Turner syndrome occurring in 1 to 5,000 newborn females. As we can see here, the Klinefelter syndrome, having additional Y chromosome is a part of numerical change in sexual chromosomes, aneuploid. The aneuploid autosomes. We have studied the trisomy 18 Edwards syndrome, additional chromosome to female or male with Edwards syndrome to chromosome number 18, causing it to have an additional autosome chromosome, not a sexual chromosome, just an autosome chromosome, causing it to have an additional chromosome, causing the total number of chromosomes to be 47 instead of 46. Edwards syndrome. Trisomy number 2, or trisomy 18. We did study the clinical features, IQ. This was the trisomy, the Down syndrome, which mental retarded with IQ scores range from 25 to 50, very low. Having a sense of it, there's the Down syndrome or Darwin syndrome, whatever it's called, Darwinism, Downism. Another autosome is the Down syndrome. Effects on mediators of hormone junction. This is the picture showing as the Down syndrome children. And this is number 21, autosome 21, or the trisomy 21, 47 chromosome with additional chromosome to number 21 chromosome. That's why it's called trisomy 21, because the autosomal 21st, this is the 21st autosomal chromosome, a pair of chromosome will be added. The additional chromosome will be added to the 21st pair of autosomal chromosomes of the human. This one's called the Down syndrome. Trisomy. Trisomy 21 and trisomy, of course we did say that the trisomy 18 was called Turner's, or the Turner's, or the all Down syndromes. The clinical features. Oh, trisomy 18, Edwards syndrome. So this is Edwards syndrome, which is better, of course, than the Down syndrome. 18 is Edward, 21 is Down. This is all the Down's syndrome, 21, trisomy 21. Or there is a problem in the 21st pair of chromosomes in a human, an additional chromosome to 21. All of those are numerical problems with chromosomes called aneuploid or aneuploidy. Trisomy 21, trisomy 18, and trisomy 13, petos. The polyploidy or the polyploid, numerical abnormalities. There is polyploidy, diploidy, or diploid, triploid, tetraploid. But we only studied the triploidy, with an additional chromosome to the Down syndrome and Peto syndrome. That was the triploidy to Down syndrome and Peto's syndrome. Also the Edwards syndrome. This is a review. Non-adjunction, failure of paired chromosome of sister chromatin passed to poles and spindle the anaphase of meiosis and mitosis. This is a non-disjunction. And the causes of non-disjunction: chemicals, radiation. Aneuploidy, trisomy +1, monosomy we haven't studied, and tetrasomy we also haven't studied, but the most important one is the trisomy, Down syndrome, Edwards syndrome, and Peto's syndrome. As here we can see the Down syndrome with a male having 47 chromosome, an additional chromosome to the pair of chromosome on the 21st pair. Monosomy, -1 chromosome. The monosomy, -1 chromosome. As we can see here, the monosomy with the 45X male, or X0, without the Y, without the XX here in the monosomy. Tetrasomy, we have studied this at the end of this lecture with an additional 6 chromosomes. The higher number, the worse the case. Euploidy, polyploidy, complements. We only focused on the aneuploidy, numerical abnormalities. This we have begun with studying the numerical and structural abnormalities. common cytogenic diseases, chromosomal abnormalities. Here we have also genetic disease with mitochondria, causing liver, heredity, optic, neuropathy, loss of central vision, cause of genetic problem in mitochondria, which could be altered and in the future maybe altered and end this disease and not for the child not to get it. More genetic disease, somatic cell genetic disease, result from altered genetic material in somatic cell like cancer. This is somatic disease. Or medical genetics, diseases, diabetes, etc. Alzheimer, genetic diseases, somatic diseases. Obesity could be classified as a multifactorial disorder affected by the environment and the genes, like obesity. Diagnosis and treatment, chromosomal lysis, no treatment with this disease. Addition to number 13, Peyto's syndrome, an additional chromosome to the 13th pair of chromosomes, autosomal chromosomes. In chromosomes disorder, could be missing. This was cystic fibrosis with the deletion of phenylalanine. Could be seen here with cystic fibrosis, heredity disorder, lung congestion, malabsorption of nutrients by the pancreas. Cystic fibrosis. Fragile syndrome, sickle cell disease, or single gene disorders. We only studied cystic fibrosis, but we know what is the meaning of sickle cell diseases and fragile syndrome. Single gene disorders, mutation in one gene. This is classification of gene disorders. Single gene disorder, chromosome disorders, multifactorial, somatic, and mitochondrial. And here are the characteristics of gene disorders: congenital, population disorder, familia. These genetic disorders or genetic disease, what's genetic disease? Abnormal mutations could be subtraction or addition. Genetic therapy and consultation is important. And what is genetical or medical genetics? Some important aspects like consulting, diagnosis, treatment, analysis of molecule, mapping, studies of inheritance. This is about medicine and heredity, medical genetics, diagnosis and management of heredity and disorders. Basic concepts: homozygous, recessive homozygous, dominance, heterozygous, genotype, and phenotype. Other homozygous and heterozygous, different alleles, and we know what is alleles and what are alleles. Dominant alleles, effect of dominant allele masks the weak recessive allele. There are many famous people having dominant and recessive alleles in the world. Number of genes in human, 2,500. locus the location of the gene on the chromosome. Basic concepts, genes and etc. This is the eukaryotic cell. We've studied it very carefully. This is centromere, chromosome, etc., etc. Genetic concerns, molecule, very important lecture. So we have studied today genetic, medical, genetic. That is very nice and important lecture that should be studied one more time and many, many times, every day, every night, every week. And we should advance from here to the next problems in the human body and genetic problems that facing the world these days and the future and how they develop. How do we develop genes, make them unharmful to the next generation, try to solve the problem of the Down syndrome and many other syndromes caused by the X and Y genes, somatic and sex chromosomes, a male and female which could cause abortion and could cause many problems in the future, the past. Thanks for listening to this lecture. Please repeat it carefully and understand every single word, like the Cry-Du-Chat syndrome, which is a 46 with an XX, that is female 5P, a missing, or this is a correct number of gene, but with a 5 minus structural abnormalities, terminal deletion, which could have many characteristics like wide set of ears, very important serious diseases caused by the deletion of chromosome parts, structural abnormality that must be studied, and what are the losses and if they are important or not losses. The deletion, and this is some. This page is very important, or was very important, that we studied molecular cytogenetics with fish and molecular genetics, analysis PCR, etc. That we should have researched online on Google maybe to search for PCR and Prader-Will to get full mark in the exam if there is an exam or a research paper. Duplication inversion should be studied again and understood very well within many diseases. And structural abnormality is insertion. Thank you for listening to this lecture, and hope you are getting more education. See you in the next lecture. Maybe it will be about another subject in dentistry or in medicine, but it will be important, of course. And let me sign out now. I'm trying to sign out. I'm trying to go offline to end this live stream.